As an island nation with a temperate climate, our green fields have ensured that Ireland is famous for its farming and food industries. Now, Irish investigators are taking advantage of emerging technologies in order to try growing crops in innovative ways.
Plant breeding is part art and part science, and the art part of it will always remain the same, but a lot more technology is available now in terms of molecular techniques and genetic techniques to identify traits of interest in varieties that can make selection more efficient in the future.
New technologies and dedicated research have also opened up new horizons for plant growers in areas like human health and are making substantial advances in the fight against some fatal diseases.
Plants are a really important new system for producing large amounts of valuable pharmaceutical proteins. And we're really excited to be at the forefront of the development of the chloroplast production system that could be used to assist in the development of AIDS vaccines.
The potato has played a vital role in Irish history. It's the fourth most consumed food in the world. And its nutritional value and high yields will ensure that it plays an increasingly important role in developing countries throughout the world. Potato breeding is an evolving science, which is helping to create new varieties with a range of traits to suit an ever-increasing market, both at home and abroad. Denis Griffin at Teagasc is enhancing traditional breeding techniques with the help of cutting-edge genetic technology.
Teagasc has had a long history. The first breeding programme was started here in 1963 by Harry Keogh and Leslie Dowley, and that programme has produced up to 35 varieties to date. So if you allow a lead-in time of 10 years for the production of a new variety, it's been one variety per year since then.
It's a lengthy process to successfully develop new breeds of potatoes, and they can often be susceptible to a range of diseases, such as potato late blight, which can have a devastating impact on yields. Here at Teagasc Crops Research Centre in Oak Park, Denis devotes his time to the breeding of new disease-resistant potato varieties in his innovative breeding programme. Each year, an exhaustive process begins.
This is the beginning of the entire breeding programme. All the genetic material and the traits that we want are contained within the parents in this glasshouse. We perform interspecific crosses here between parents with complementary traits. We take flowers before they mature and emasculate them or remove the male anthers. This prevents the flower from self-fertilizing. Then we select a male parent with complementary traits to the female parent. We remove pollen, place it onto the female stigma of the flower. If the fertilization is successful, a berry forms. So if a cross is successful, a potato berry is produced, and this is just what the seed looked like in a berry. We can get up to 200 individual seeds in a berry, and each one at this stage is a genetic individual, but a sibling. We extract the seed over the winter months. Each individual seed here is a potential new variety, and it can take up to 12 years of selection of these and growing of these seedlings to produce a new variety. This is year 2 of the potato breeding programme. This is where we plant all our seedlings that we've seen from the crossing house. Each plant here has been produced from one of the true seeds that we show, and each plant here represents a potential new variety. This is one example. We've moved from true seed to a tuber generation, and you can see that each plant produces 3 to 4 small tubers. Because we have very little seed. We can do no testing in terms of disease resistance or cooking quality or anything like that. We have to wait until we multiply more seed. So we take one tuber from each plant, and these are sown in the field in Wicklow, in our isolation centre in Wicklow, and we begin to select the numbers from there over about 10 to 12 years.
Out of the 200,000 unique seeds produced in the first-year breeding programme, the number of emerging potato varieties is whittled down to 1 or 2 over a 12-year period. Denis and his team then begin to hone in on the seedlings that contain the ideal combination of traits that will ultimately determine the success or failure of the new variety.
Okay, we're here in Broadway, County Wexford today doing an early potato trial. This links into our potato breeding programme from about the year 6 to year 12 stage. After the early stage selection, which we've seen, we've narrowed down the number of seedlings to approximately 60. What we try to achieve here is we look at the potatoes on the field to determine how suitable they are for the market. Potato is a very, very difficult crop to select. There are over 50 different traits that we assess in the production of a new variety, and it ranges from all the cooking and taste characteristics, disease resistance, shape, right through to maturity, flesh colour, texture. It's very difficult to combine all of those traits into one discrete variety.
One of the biggest threats to the success of any breeding programme is the susceptibility of those varieties to disease. Blight and pest-based disease are still very much a cause for concern for Irish farmers. Helping Denis to identify and get to the origins of these and many other traits, one Teagasc investigator, Dan Milbourne, is using a very special technique, genetic fingerprinting.
Denis has the, I suppose, unenviable job of selecting varieties based on the expression of genes, the characteristics or traits. My part of the process involves tracking down the genes that are responsible for these traits and giving Denis the capacity, instead of having to select on the basis of the expression of the gene, which is what you see in the field, give him the capacity to look directly within the plant, and I can tell him for definite whether that plant has the gene he's interested in. This is a genetic fingerprint of essentially 31 breeding lines in our breeding programme. Now, people will be more familiar with the concept of genetic fingerprinting from programmes like CSI and things like that, where they're used in a forensic setting. But we use the exact same kind of technology here in the breeding programme to try and make the process more efficient. You can read a genetic fingerprint basically as a sort of molecular barcode, if you like. Each genetically distinct individual will have its own barcode pattern. And each of the bars, or as we call them here, bands, that appear on this autorad is essentially a piece of DNA which is either near or is a gene. If you look at enough fingerprints, what you begin to notice is that certain bands on the gel are associated with traits that we're interested in. That becomes a sort of diagnostic marker then for that trait. I don't know, I think we'll do occidentally separately.
As Denis identifies new varieties, Dan can identify disease-resistant genetic traits at a very early stage, making the process much more efficient. This new perspective on the potato varieties is vital in an increasingly competitive and changing marketplace.
As the marketplace changes very quickly nowadays, we see, for example, with climate change, we're getting higher temperatures, we have maybe less rainfall. So as things shift, we need to shift the breeding effort to try and produce material to grow in those conditions. The other aspects we're facing now, for example, chemicals have been withdrawn, chemicals we use to control blight and to control pests. The list of chemicals available to farmers is reducing all the time, so resistance to pests and diseases is becoming more and more important. The armoury the farmers have is becoming smaller, and we hope to build more resistance in to PCN, to blight and so on, you know.
This is some late blight which has infected a potato seedling. And in crops which haven't been sprayed, the loss can be 100% if you get an early infection in the year. Our climate is particularly well suited towards blight. Now, resistant varieties would have a lot to contribute in that context, in that varieties would last longer without fungicide applications, but also would need less applications of fungicide. The last 2 years have been particularly difficult blight years due to the weather conditions.
Using genetic fingerprinting, Milbourne is now able to identify disease-resistant traits for blight and the potato cyst nematode, or PCN, by the presence of DNA markers, which maximises the chances of selecting new resistant varieties in the future.
So we've got something here. If you look at this particular one, there's a band here in this individual which is associated with resistance to potato cyst nematode or eelworm, which is a very, very important pathogen, which is one of the breeding targets for the programme, to breed resistant varieties to this. It's a soil-borne pathogen. It's a microscopic worm that infests the roots of potatoes. And the way it's controlled currently is to actually take very, very toxic substances and rotivate them into the top several centimetres of the soil. And there's concerns about using these chemicals to control them. So developing varieties that are resistant to PCN is a very, very important target for breeding programmes worldwide. Here we've managed to develop a genetic fingerprinting assay that allows us to select from the breeding material individuals which have a gene that confers a very, very strong level of resistance to PCN. And so this allows us to give Denis information about individuals in the breeding programme before he could otherwise actually get that information using a traditional disease resistance test.
One of the limiting factors in the production of varieties has been the amount of labour it takes to screen varieties at a very early stage, and the amount of potato material which is available to us, because most disease resistance tests are very destructive and use up plant material. By conducting these genetic tests at a very early stage, we can identify the genes and hence the traits of interest and retain those in our programme and increase our selection intensity for those traits. And this maximises the chances of us producing more disease-resistant varieties in the future.
In Oak Park, at the tasting stage of the programme, Denis gets closer to finding those varieties that will be seen on the supermarket shelf, both here and abroad. As Dan's work on the genetics of the potato progresses, the relationship between breeder and geneticist looks likely to yield even more exciting results.
So at the moment, we're working in what we call an incompletely characterised genome. Teagasc is involved in an international consortium of about 15 laboratories worldwide, whose goal is to find the entire genome sequence of potato, much in the way as has been recently done with humans. And that sort of information is going to make this sort of genetic fingerprinting application within breeding programs much, much easier, much, much more commonplace and much, much more powerful. And we'll be able to do it for more traits than we can at present.
These new technologies are never going to replace entirely plant breeding, and I think there's still a necessity for the breeder's eye, but they're going to make the process a lot more efficient into the future.
Over the years, Irish people have used their own land to nourish themselves and their families. In much the same way, Irish investigators are looking to certain crops to see if they can harvest their inherent health-giving properties.
The focus of my research really is to try and increase levels of compounds that are already present in the food that have beneficial properties. So just enhancing the levels of beneficial compounds in the food itself.
Dr. Nigel Brunton is looking at the health-giving properties which naturally occur in certain foods. One of those compounds that interests Brunton is beta-glucan.
Beta-glucan is basically an insoluble fibre. It consists of a long chain of sugars. And the reason why we're very interested in beta-glucan is that it has been shown to have a lot of properties beyond basic nutrition. For example, it can promote immune function, it can have a probiotic effect. It can also lower cholesterol, and that's the reason why we're actually trying to enhance levels of beta-glucan in a bread. We already knew from other people's results that barley had high levels of beta-glucan in it. So the first step into producing a bread is to actually mill the barley grain. During the milling process, you generate a number of different fractions. For example, you produce a flour, you produce a bran. What we were very interested in is to look at the levels of beta-glucans in these different fractions.
Brunton's research threw up some interesting and unexpected results.
The exciting thing, I suppose, is that we found 4 times the level of beta-glucan in the barley end fraction, and this is the fraction that is normally thrown away, so therefore we're actually increasing the value of the barley while also increasing the nutritional properties of the breads that we were going to incorporate the barley ends into. So the next challenge then is to take that fraction and incorporate it into a bread. And what we want to do is to produce a bread that tastes as good as normal breads that are on the supermarket shelf, which has higher levels of this helper-promoting compound in it.
For normal white wheat bread, people are very used to its taste and used to its flavour. So when you're replacing wheat flour with something else, it's always quite difficult to try and get something that will simulate wheat bread completely. When you're making bread, you want to get a viscoelastic dough, a dough that you can stretch and knead into the form of a bread loaf shape. And it's the gluten in wheat flour that is responsible for this property, and therefore it's very difficult to try and get an elastic dough if you don't have the gluten present. So that's what we're trying to do at the moment. Okay, well, this is a normal wheat bread that you would find in the supermarkets. It is 100% wheat flour formulation, and these two breads would have different levels of barley inclusions. This would have 15% of barley germ. This would have a much higher level of barley beta-glucan, and as you can see, there's an optimal level before the product quality will start to decrease. So what we're doing at the moment is we're trying to find the optimum level of barley inclusion without detrimentally affecting the quality of the breads. Well, I think that the functional food area is going to grow a lot. I think that bread, because it's such a universal product, it makes for a very easier carrier for healthy ingredients. Research is starting to increase at the moment now in the area of nutritious cereal products, so definitely in the future it's an area that's going to expand.
While scientists like Nigel Brunton continue to examine the naturally occurring health-giving properties of food, others are attempting not to exploit the inherent compounds of plants, but to use them as factories to grow foreign cells inside them. For centuries, scientists have been looking at plants for their healing properties. Today, investigators are looking at plants in a new way, which may prove helpful in the fight against a range of diseases. Dix of NUI Maynooth is working to change the genetic makeup of key plant cells, results of which might play a critical role in human disease such as HIV/AIDS.
Well, plants have always been a very important source of medicines. There are many, many examples of a wide range of plants. And this is an example behind me, in fact, of a medicinal plant. This particular yew tree is a very famous one. It's probably more than 1,000 years old or thereabouts, possibly the oldest in Ireland. And it's known as the Silken Thomas yew because, reputedly, Silken Thomas, the 10th Earl of Kildare, played the harp under this on his last night in Maynooth before his rebellion was finally crushed by Henry VIII's forces. Well, in the 1960s it was discovered that the bark of this tree contains a chemical called Taxol, which is very effective against a range of cancers. The chemical is produced in the bark. If you remove the bark, you kill the tree. And it's also produced in relatively small amounts, so you have to sacrifice a lot of trees in order to treat patients. Now, with the new genetic technologies available to us, we can start looking at very different medicines, ones that aren't necessarily of plant origin at all. For example, we could look at the production of human insulin, or antibodies, or vaccines. There's the possibility of using proteins produced in plants to help in the efforts to produce a vaccine against AIDS.
Dix is looking at plant systems as a way of growing key proteins involved in the HIV disease mechanism. This is a revolutionary new system by which a plant is selected as a form of surrogate host within which it's possible to grow a completely foreign compound, such as this key protein. Traditionally, the methods of producing these proteins were in bacterial or animal cell cultures. However, these are extremely expensive, and because they run a serious risk of contamination, they have to be maintained in highly sterile conditions.
Plants have a big advantage over these systems for several reasons. Firstly, you can grow up large amounts of plants, a large amount of material in a confined space very cheaply. In addition to that, There's no risk of infection with animal viruses that you can get in animal cell cultures, or the production of toxins that you associate with microbial cultures. We use tobacco for this work because it's a model plant for the plant tissue culture procedures. It works very well for these methods we have to employ. But in addition to that, it's obviously a plant which produces a lot of leaf material, which is where we're producing the protein of interest.
In HIV, the virus intercepts the human cell. Inside the virus, the RNA, which contains the viral genes, begins to replicate and infect the cell. The P24 protein surrounds and protects the RNA. With the replication complete, the virus then exits the cell and begins targeting other healthy cells. If antibodies can target the P24 protein and knock it out, it will destroy the virus's capability for protecting its harmful genes, thus exposing it to potential vaccines.
So the idea is to introduce the gene that's responsible for making the protein of interest, the HIV protein, into the plant cells so that we can produce the protein throughout the whole plant. And, of course, if we then collect seed from that plant, we can grow up greenhouses full of those plants to produce the protein.
Dix's approach was novel. He bypassed the nucleus and targeted another site where the genetic material was plentiful, the chloroplast. The chloroplast has its own DNA molecules, up to 1,000 copies in the cell, as compared with just 2 in the nucleus. So when a foreign gene is introduced, it could be amplified up to 1,000 times, producing large amounts of the foreign P24 protein, making it a much more efficient production system.
Another attraction is that we can be very precise in where we insert the foreign gene into the DNA molecule that the chloroplasts have, in contrast to nuclear transformation, where that insertion takes place at random in the nucleus. This means we can avoid interfering with any other gene functions. In order to do this, we first of all grow some small sterile plants in tubs. We remove the leaves from these and place them in a special chamber in what we call a gene gun. This enables us to fire small particles of gold coated in our DNA, which includes our gene that we want to introduce. We fire those at the plant material. The DNA penetrates the plant cells, and some of it will end up in the chloroplasts. This can become incorporated into the chloroplast's genome, and gradually that new population of chloroplast DNA molecules can take over until all the DNA molecules within the chloroplast, and then within the cell, contain the gene of interest.
Having shown that they could grow the stable HIV protein in large amounts in plants, the next stage was to make sure that the protein was as effective in the new plants as it was in the bacterial models.
So we had actually a PhD student who came here for a few weeks and extracted the protein from the leaves, and she took the protein back to George's Hospital, and she's been injecting mice with an E. coli version of the protein and our plant-made version, and they found that the mice respond in the same way to both proteins, so our plant-made protein is as good as the E. coli protein.
Dix and McCabe's work is attracting significant interest in this new and vibrant area of plant science. Researchers from around the world have gathered in Cork for a conference on this emerging field.
The work that Phil Dix is doing in Maynooth, targeting gene expression to the chloroplast for pharmaceutical production, is probably one of the leading labs in the world, because this is still pretty young science. We're just beginning to start looking at the notion of using plants for expressing human therapeutics, and there's really only a few leaders in the world. And I think Phil Dix is really in a position to be ahead of the curve, because he's one of the first to actually look at the notion of using chloroplasts for expressing human therapeutics. So Europe, and Ireland especially, I think could really make a name for themselves in this field.
I really believed and still believe that it's going to have enormous impact in the developing world. We're going to produce a variety of the new plant-based pharmaceuticals. It's going to benefit people in the developing world as well as, of course, the first world. But I think the big potential is taking high-tech medicines into low-tech parts of the world. These are the sorts of things I think that Ireland has been very good at adapting, taking new strategies for technology and implementing it in a high-tech way.
Well, we've had great success with the production of this particular HIV protein, P24, and we're hopeful that we'll be able to extend this to a broader range of diseases in the near future.
The use of plants as production systems for pharmaceuticals is gathering momentum throughout the world. It's clear that Irish investigators are delving deeper into the genetic and molecular makeup of crops and plants in a bid to better understand how to harvest the potential within. Although this is still a relatively young science, results to date indicate exciting possibilities that should bring us all significant benefits into the future.